thank you everyone for showing up and for showing your interest in trying to generate some 3D genome models using Chrom3D. The goal of today's tutorial is actually to make what I've tried to show you here in a video, where at the end of today's session, maybe in a couple of hours, we should be able to use the Chimera X program to interact with a model of, well, what we're going to study today, which is human chromosome 18, which is what you can see here. In this visualization, the red regions correspond to regions associated with the periphery of the nucleus, the so-called nuclear lamina, and the gray regions correspond to everything else on the chromosome. We will go deeper into the details there later. But this is what we're trying to do, and we're going to use Chrom3D to generate these models and this Chimera X system to visualize and kind of interact with the model. So today's tutorial will actually use a GitHub page that I'll give you the link to later. There is a full genome version of the tutorial that I'm going through today. This you can go, you know, if everything works fine for you, you've installed all the programs and you can easily manage today's tutorial, I'll recommend for you to go in and try the full genome version, where you can actually model the entire human genome. But that's on a separate GitHub page that you will get the link to later. This is an overview of today's steps of the tutorial. Basically, we're going to use some data from LADs in a bed file format and raw reads from Hi-C, so starting with FOSQ files. Before I go into the details of the overview of today's tutorial, to make sure that we're all on the same page, I know many of you will know about these techniques already, but let me just make sure that everyone is on the same page. So we are going to use data from this Hi-C technique. In this technique, the goal is to try and identify pairwise contact frequencies between different regions within and between chromosomes. And the way the technique works is that you start by cross-linking DNA, meaning that at the time point of cross-linking, regions that are close together within the nucleus will be physically bound to each other via the proteins that bind to these. So for example, in this visualization, this blue piece of DNA and red piece of DNA was close together, and upon cross-linking, you can see them binding together. If you then cut these structures with a restriction enzyme, you end up with lots of these protein complexes with pieces of DNA sticking out, and these pieces of DNA were the regions close in the nucleus, and are the regions we try to capture. In the next step, we can fill in the ends and mark with biotin, induce ligation, so you get this kind of circular structure. If you then purify, share the DNA, and pull down on the biotin mark, so that mark will be at the ligation point between the two pieces of DNA, then you enrich for these regions, and using paired end sequencing, you can then quantify by mapping back to the appropriate reference genome, quantify the interactions or the pairs of regions interacting genome-wide, basically. And in this visualization here, I'm just showing, for example, in human chromosome 6, you can visualize these data as a heat map with kind of white color as indicating low number of interactions and red color indicating high number of interactions. And already by inspecting, for example, the contact patterns on chromosome 6, you can see enrichments and depletions of contacts across the chromosome. If you zoom in to the diagonal, you will see further structure, these topologically associated domains that have now become very famous. We will use these TADs. We will actually divide the chromosome into a segmentation of the chromosome, basically, based on the size of these TADs. So we will go into that later. Also, when you inspect contact frequencies across chromosomes. So in this visualization, I'm looking at interactions between chromosome 6 and chromosome 7. You will also see enrichments and depletions of contacts interchromosomally. But in today's tutorial, we'll focus on chromosome 18 interactions. We're also going to use data from lamin B1 ChIP-seq. So lamin B1 is a protein that can be found more or less exclusively in the nuclear periphery, coating the inner nuclear membrane, and is part of the so-called nuclear lamina, which is a protein meshwork at the inner nuclear membrane, and chromatin that interacts in the periphery is typically repressive. So actually, to identify the regions in the genome placed out here in the nuclear periphery, we can use ChIP-seq, so chromatin immunoprecipitation, to enrich for regions that are associated with this lamin B1 protein, purify the DNA and sequence the DNA and map it to the genome, and thereby producing these sort of histograms that quantify then where across the chromosomes these regions associate with the protein. And from these profiles, these histograms here, you can generate a segmentation using a peak caller. Essentially, you can then generate a bed file, basically, giving you the positions of these enriched regions where the genome is interacting with the nuclear lamina. So what we're going to do in today's tutorial is we're going to use a bed file consisting of these LADs, again on chromosome 18. We're also going to use Hi-C data in fast, so basically raw sequence reads in FASTQ format. These will be processed with the Hi-C Pro pipeline. This will map the reads to the reference genome and aggregate and quantify the numbers of interactions across the chromosome that we're going to look at today. So I didn't mention this, but the reason why we're looking only at chromosome 18 is for to make it fast enough to go through in today's tutorial. Then from the output of Hi-C Pro, we will use a program called Armatus. This is a TAD caller. So this will identify these topologically associated domains, or TADs. And these we will use actually to segment the genome and to define the position of the beads, essentially. Then, using the Hi-C data coming out from Hi-C Pro, we will aggregate the interactions between all the beads. Then we will use a statistical program based on the so-called non-central hypergeometric distribution to identify significant interactions in this Hi-C contacts between the beads. Finally, these will go into a file together with the definitions of the beads and the LADs, a so-called GTrack file. We will take a look at that later. And this is the input file that goes into Chrom3D, which defines, well, where the beads are, which beads should be restrained towards the periphery, which beads should be close to each other. And basically, we will run Chrom3D to generate a model that we can visualize using Chimera X. So that's the plan for today. So now I will ask everyone who wants to participate synchronously while I'm going through the steps to go into this GitHub page. Then it will look something like this here. You can read this. I'm not going to go through everything here. There's information about what the data that we're using today, where they come from, and so on. There's some more details and links to the various tools that we're going to use, and some more information. There's a list of required software that you need to install before you can run all the steps. So in a short while, we will start with the tutorial. Before I do that, I'll explain how you can jump in at various points in various steps in the tutorial, even if you get stuck in some of the places or if you didn't manage to install some of the programs. At various places in the tutorial, you can see an orange line where it says, if you're stuck at this point, and so on. If you copy the data from, or the commands from just under that orange line, it means you can jump on to the next step. And there's a bunch of these places where you have this orange line where you can jump in. Everyone has to perform, well, you have to set up the tutorial. I'll do that right now and ask everyone also to do the same. You can just copy these two lines. If you're running this on a Mac, you should be able to run the tutorial on your Mac. If you're on a Windows computer, you should be using PuTTY, for example, to log into an external server. Or if you have a Linux computer, you can just also run it on your computer. So what we've done here is just downloading today's tutorial. This includes some files. I'll actually show you here in my terminal. We download a folder called Inc Tutorial. In here, there's a number of files. One of them is the README file, which is just the same file that we're looking at here to the left on the GitHub page. There is a folder called Backup, and in there is actually the data that allows you to jump in at the various points during the tutorial. There is a FASTQ folder directory. In there is another directory called Chromosome 18, and in there you have the FASTQ files from the Hi-C data that we're going to use today. These have been downsampled to only include reads mapping to chromosome 18 to make it possible for us to run through all of the steps here today in a reasonably short time. Then there is this folder, which contains a tarball zipped, which has the bow tie 2 indexes for chromosome 18 in there. We'll look at that later. And finally, there is a folder called images, which just contains 2 images that we're going to look at the end. So with that, we can perform the second step. This one creates a new folder for us called LAD. We will download a bed file from the internet. These are already called LADs from the same cells for which we have Hi-C data. And then we will unzip the file. I'll just show you. You don't have to do the same as me now, where I will just show you what these LADs look like. It's a bed file, so giving the chromosome start and end positions of the LADs, and also a score that gives an indication of the quality of the calling algorithm on the LADs, basically. I will just proceed now to step number 3, where we will download some files that we need for running Hi-C Pro, essentially. The first file that we download is a configuration file, which we will modify a little bit before we run Hi-C Pro. Then there is a file containing the size of each of the chromosomes on human genome version 19. And then there is a file called hint3 resfrag_hg19.bed, which is a bed file consisting the restriction fragments on using the hint3 restriction enzyme, basically. I'm showing it to you here. This is just another bed file giving the chromosome start and end position of all of the restriction fragments on human genome version 19. Yes, and then we unzip the bow tie 2 indexes into this folder. You can see them here. So far, so good. I'll move on to step number 4. then you can again just copy paste these lines like this. This will make another folder for us called processing scripts. We download into this folder some scripts from the Chrom3D package. Then into this folder we unzip some scripts that we're going to use later, including to run the statistics, to make the G-track file, and so on. So this is just setting up some files that we will need in later steps. Now that that's done, I'm going to run the next step. So that's step 5, which unzips this program called NCHG, which is the statistical program that we're going to use. And it compiles the program for us as well. This is something that we're going to use later. Then then we will actually go into the configuration file for Hi-C Pro. So what I've also written here in the text is I ask you to use your favorite text editor. For me, it's Emacs. You can use other types of editors in the command line, or if you're on the Mac, or you can use text edit, for example. But I'll just use Emacs for now to modify, to open up and modify this file called config Hi-C Pro, which we have downloaded. So before we actually go in there, I'll ask you to type pwd and copy this path to your current working directory. We're going to use that once we go into the configuration file to modify one of the lines. So I'll just go into the configuration file now. And in the tutorial, it says that we should go to line number 39. I can see here which line I'm at. So at line 39, we are asked to provide the path for the bowtie 2 indexes. So I'll paste in the full path of my working directory. And then we have to give as said here, the name of the actual bowtie 2 index that was provided for us in the GitHub. So so we have to write HG19, chromosome 18, and /. This part will be the same for all of us, but of course, where you have your downloaded this tutorial, where you did git clone, will be different. So this part will be unique for each of you. But this is now the first modification that we're going to use due to the file. Then the next modification is on line 47. So that's here, just the name of the reference genome. When you're running the full genome version of this tutorial, you're using HG19, but in this case, we're using a downscaled version with chromosome 18 only. So this is called HG19 _ chromosome 18, like you can see here. And finally, you should go to line number 89. Which provides the different bin sizes of the Hi-C data that you want to compute or aggregate the contacts over. I will ask you to delete this line completely and type in 50,000 and 1 million. Like that. And that's basically it. So when you use Hi-C Pro for your data, you will have to modify the lines I showed. But of course, if you use a different restriction enzyme or a different bowtie index, a different reference genome, then you need to modify it accordingly. I'll save the file now and go out. So now we have modified the configuration file so that we can run Hi-C Pro. And the next step is going to take 10 minutes to run Hi-C Pro. So I'll just ask you to copy it in and start running it. This step takes the configuration file that we just modified. It takes the FASTQ files. They are in the FASTQ folder, and it will output all of the results into a new folder called Hi-C Pro results. And while this is running, I'll allow you, of course, to catch up. If you are completely stuck, you can always run the commands written under the orange line. So while my Hi-C Pro is running now and mapping all the reads and aggregating the contacts across chromosome 18, I'll explain a little bit more about Chrom3D and the input to Chrom3D. So what is Chrom3D actually? Chrom3D is a program that tries to make these 3D genome models. It was published by or developed by me and others and published a couple of years ago in 2017. The way it works is you basically define a beads on a string model for each chromosome, where the size of the beads is defined by the user. In today's tutorial, we are going to use the TADs, and you will see that some of the beads are bigger, and those correspond to larger TADs, basically. Between the TADs or the beads, we will be able to define various restraints. So we can, for example, define interactions between beads based on the Hi-C data that you can see here, based on significant interactions between these TADs. We will also be able to define beads associated with LADs that should be restrained towards the nuclear periphery. So Chrom3D actually sets up a model of the nucleus, and the chromosomes will be confined within this model of the nucleus. And through randomly selected alterations or moves on the chromosomes that you can see in a video here that just shows from a full genome model using Chrom3D what these moves look like. You will start with a random configuration of the chromosomes like you can see here, and gradually you will optimize the loss score, and the loss score incorporates all of the restraints that you have set up prior to running Chrom3D. So during the assimilation run, this loss score will be lower and lower towards a converged structure. This is what we're going to do today. And at the end, you will see the optimized structure. In this case, this is a genome-wide model. This can now be placed within a model of the nucleus to allow us to visualize it like this, where each chromosome, in this case, is colored in a distinct color. And you can see the different beads, which corresponds to the different TADs. The inputs to Chrom3D is in a so-called GTRAC format. This is not a format familiar to many people, I know, but this is more or less the same as a bed file. The first 4 columns in the GTRAC format is actually identical to the bed format. So here's an example of a kind of toy data set in a GTRAC format. The first column gives the chromosome, then the start and the end, and then the 4th column gives an identifier, a name basically for this segment. So this will typically be the TADs, but it could also be other ways of segmenting the chromosomes. Then the user can add additional information to setting up the restraints of the model. For example, if you add a column with the name periphery, if you specify a 1, it means that this TAD or this bead will be restrained towards the periphery. If you provide a dot in the same column, it means that this TAD will not care whether it's in the periphery or in the center or anywhere else. If you give a 0, it means that this TAD would like to go to the nuclear center. In the same way, we have a column called edges, and in the edges column, you will give interaction restraints. So for example, In this case, I want TAD1 to be close to TAD4, and TAD4 should, in addition to be close to TAD1, also be close to TAD6, and so on. And these interactions are separated with a semicolon. So basically, you can visualize it like this. This GTrack file sets up how the TADs are connected to each other in the beads on the string chain, their size, and how they should interact with the nucleus or the nuclear confinement, the nuclear periphery, and the center, and also how they should interact with each other. And finally, you can add, for example, a column called color, which gives the color of the bead. And it's set up so if you skip one of the, let's say you skip the periphery column, Chrom3D will still run. It's just that then there will be no constraints or restraints relative to the nuclear periphery. In the same way, if you don't specify colors, Chrom3D will select random colors for you instead. There are additional options you can add. We're not going to do that today, but I just want to let you know that you can add weights to the different constraints like this, and weights also, for example, for the interactions, as you can see in green here. But this is not something we're going to do today. So what has actually been done now is that Hi-C Pro has mapped the reads and aggregated all of the contacts between the bins that we have defined. So after you've done this, or if you actually managed to run Hi-C Pro, the next now is for us to make some folders. So we will run the commands in step number 8, like this. We just make some directories, one called bedPE, and within it intra, and then one called matrix. So now that we have these, have this Hi-C folder, and within it the bedPE and the matrix folder, we will convert the output from Hi-C Pro into bedPE format and matrix format. The bedPE format, I'll start by performing all of the steps by copy pasting these in and show you. So the first line here, actually, this is a long one-liner that basically converts the data from the Hi-C Pro output into a different format, basically, that aggregates all the contacts for each of the bins and sends it into a new file named like this. I'll just show you what that looks like. So this is the bedPE format, gives the chromosome position, start position, end position, and then a new 3 columns, which is the interacting bin for this particular line. And this column here gives the number of interactions from the Hi-C for these different pairs of bins. So that's relatively simple format that we're going to use later. In this other one, the matrix format is basically the similar information but organized in a different way. Here you can see a 0, which just indicates the starting position, and here's another 0, and the name of the file actually gives the information about the chromosome. But again, we have the numbers of interactions corresponding to each of the pairs of bins here. This is the format needed for running the Eukaryotes program. We will now perform step number 10, which is running the Eukaryotes program to actually call the TADs based on these Hi-C data formatted in this way. I'll start by making a new folder within the Hi-C folder called TADs. These are where the TADs are going to live. And then we will run the command actually running Eukaryotes. Just copy and paste. If you encounter issues here, I know depending on how you have installed Armatus on your system, if you get an error message that it cannot find Armatus Linux x64, it could be because you have installed it differently, and on your system it's just called Armatus. So if you get an error message, try to run exactly the same but skipping these things here. We have a question now regarding the TAD caller. Is it possible to use other TAD callers than Armatus? That's absolutely possible. We have used Armatus in our system because it seems to work quite well. The only thing with Armatus is you have one parameter here, which is this gamma max parameter, which you have to kind of play around with a little bit. It basically sets the, so as you know, the TADs are nested, and there is no clear definition on which level of nestedness that makes sense in general. But for your favorite data set, you might have what you can basically do is you can run Armatus with different settings for this gamma max parameter, and basically pick the level of nestedness that you think is appropriate for your particular purpose. So in our hands, this Armatus, if you can manage to get it installed, works really nicely to give TADs at a decent level. And then there's a question about what criterion I use to actually select this parameter. I think the best so what the criterion we have used is actually looking at the size of the TADs. So since this is used to kind of determine the level of nestedness, this will be related to how large or how small the TADs will be. So depending on which organism you're working on or which system, you can try out different settings for the gamma parameter and look at the average size or the distribution of the sizes of the TADs to see if they make sense. You can of course also visualize it, so to look at the Hi-C data together with the segmentation of the TADs to see that this level makes sense for my particular application. Now we will run step number 11. This is just a simple step which will convert the output from Armatus to, which should now be in the, so the output from Armatus is in the Hi-C TADS chromosome 18 consensus .text. It looks like this chromosome. It looks like a bed file, but it's not exactly as a bed file. After running step 11, you will actually make the bed file, which just adds a 1 to this column number 3. So Armatus is end inclusive in the output, so that means that includes the last one, the last position here, but that is not the case for a bed file, so therefore I add a 1 to the last column to actually generate the bed file from this. What we'll do next, and this is an important step for Chrom3D, is that we will convert the TADs into a fully segmented genome. What you can see here from the output is that, for example, there is no TAD starting from position 0 and going up to 150,000. It starts on 150,000 basically. What we need for Chrom3D is a fully segmented genome. So starting from position 0 and all of the gaps filled, so to say. So to generate that, we will now perform step number 12. In this step, we will use BedTools complement, which will fill in all the gaps for us in between the regions where there is no defined segment. So just looking at the output of that. So what you can see here in the file that we now call chromosome 18 _beads.bed is that this has now a segment defined starting from position 0 on chromosome 18 up to 150,000. And if you just look at, you don't have to do this, I'll just show you how many beads do we have compared to how many TADs are on the chromosome. You can see that we have 97 beads, but there were only 71 TADs called from Armatus. So it just fills in all the gaps. So we have a segment, a fully segmented chromosome 18 in this case. Right, so now that we have the beads defined in this chromosome 18_beads.bed, we are able now to map the Hi-C data to the beads, allowing us to count how many interactions do we see actually between the beads that we have defined. And that's what we're going to do in step number 13. So I'll ask you now to copy this under step 13 and execute it. So what this step actually does is that it'll take the interactions in BED-PE format that we generated earlier. It will pick out the left side, so the first 3 columns. It will actually take the chromosome position, the start and the start +1. So it's the starting position of the left side of the interaction. It will use BED tools intersect to intersect those contacts in BED-PE format with the beads, and then send that into a temporary file called left.tmp. And then it will do exactly the same thing, but for the right side of the BED-PE format. And then intersect again with the beads on chromosome 18. And then also outputting the number of contacts into the file, into a write.tmp file. Once that is done, we can use paste to paste these two together. Using a hash table, we can count and aggregate the numbers of interactions between beads. So because this file here will contain repeated, so for all of the, whenever 2 Hi-C interactions cover the same bead multiple times, you will be able to aggregate them using this awk line here, basically. This just makes sure that we are looking at intrachromosomal interactions. It will not really apply in our case, since we are only looking at chromosome 18. And then it will sort the file and finally output these into this new BED-PE file that I will show you here. Again, you don't have to look at it. I'll show you what the file looks like. So this is now a new BED-PE file with chromosome start end of the bead this time, and not on the Hi-C bin. And the same thing, so this bead interacts with this bead with this number of Hi-C interactions. Okay, so in step number 14, this is a long one-liner that will download. So the point of the step is to remove any interaction that overlap centromeric positions, because these are known to be artifactual. So basically we will download from HG19 all information about the positions of the centromeres. We will unzip that file, we'll grep the centromeric positions, we'll use BED tools pair to BED to intersect our interactions in BED-PE format together with these centromeres. And with using type neither, we'll make sure that neither of the interactions, neither of the beads in the BED-PE file should overlap with centromeres. And finally, the resulting data will go into this file here, which is the same file but contains no sen, meaning that we removed all of the interactions involving centromeric regions. Again, if you now are kind of lagging behind, so if you're not at step 14 or something broke or didn't work and you want to follow along, please just copy the line under the orange line to be able to run the next step 15. In step 15, we will use this script that we compiled previously called NCHG to determine statistically significant interactions between the beads. So to do that, you will just copy these two lines and paste them in, and it should be relatively fast. The first line here uses this NCHG program. It specifies the resolution of the Hi-C data at 50 kb. It specifies also the input data set, so the BedPE file that contains all of the interactions, excluding the ones with the centromeres in chromosome 18, and output those to a new file called the same but .out. In the second step, we're using a Python script called this, which just calculates, it calculates the false discovery rate and takes as input the output from the previous step, specifies that we're using the Benjamini-Hochberg definition of the false discovery at 0.01 level, and it requires a twofold enrichment of the interactions over the expected interactions. And then it outputs that into a new file called the same but with .sig. So these are the significant interactions from the Hi-C data between all of the beads. I provided some more information about the NCHD, so the non-central hypergeometric distribution. It's a statistical model incorporating the total number of interactions on the chromosome, the total number of interactions for each of the pairs of beads, and also the genomic or linear distance between the two beads in order for us to calculate a p-value on the significance of the number of interactions between all of the pairs of the beads. Here we will generate the input file in the GTrack format for Chrom3D, and thereby specifying the setup for the Chrom3D model. So please copy these three lines and paste them in. Then, so what this does is that it uses the script called make GTrack. It just makes it takes the interactions from the significant bead-bead interactions. It also takes all of the beads and generates a simple GTrack file. I'll show you, you don't have to run this particular command. This is just for me to show you. For example, here you can see the definition of the first bead starting at 0 up to 150,000 and the name that is generated for this bead, and some information about the interactions and so on. As you can see, this file does not contain the header that we need. So that's actually what's done in the second step. It just echoes the header that we need and sends that into a new file. And in the third step, we use BedTools intersect -c to provide information of, to basically intersect the GTrack file, which remember is similar to a bed file. So we can actually use the GTrack file with BedTools. It will intersect those with the LADs that we downloaded all the way in the beginning. And then it will reformat it into the GTrack format in this one-liner here and send that information into the GTrack file. So I'll just show you, you don't have to do this step here, but this is what the end result looks like. It gives the header information. There is also some information which will be ignored by the Chrom3D, but which indicates that this is a GTrack file. And then you'll have the appropriate header for what we're going to do today, which gives the chromosome, the start, the end position, the ID of each of the beads. There is a column called radius as well. I didn't talk about that previously. For now, we set the radius of each of the beads to 1 because by default, Chrom3D will scale the size of the beads according to the size of the segments. So you can either skip this column completely or include it and specify one, or if you specify specific values, you can determine or decide yourself exactly what the size of each of the beads should be. Then there is the periphery. You can see some dots, but also some ones here. And finally, the edges column, which contains all of the interactions between the different beads, based now on the significant interactions that we determined in the previous steps. Then the next step is actually running Chrom3D on the GTrack file. So you can see now it's the Chrom3D started running. So here the parameters specifies the number of iterations. Here it's 100,000 iterations. Minus R gives the size of the nucleus, so basically the radius of the nucleus in micrometers, for example, so 5 micrometer radius size of the nucleus. This flag here, minus nucleus, specifies that we want to model our chromosome within the confinements of a nucleus. If you skip this, then the beads will not be forced to stay inside of the nucleus. This parameter, minus y 0.01, gives the volume, the percentage of the volume of the model relative to the volume of the nucleus. So for a full genome, we usually set this to somewhere between 0.1 and 0.2. So that means 10 or 20% of the nuclear volume is occupied by the chromosomes. In this case, we only have a single chromosome, chromosome 18. And I must admit, I don't know exactly how much of the nuclear volume chromosome 18 fills up. But this number should be relatively close, maybe a little bit too large on the actual volume. It's not so important, but just observe that it's possible to set this volume of the model. Then there's minus L, which gives which specifies how often we should get information in the terminal during the simulation. So now I say every 10,000 iterations, Chrom3D will output the loss score that we try to minimize, that you can see here. Minus C 0.001 specifies the cooling rate during the simulation, which also means that I'm using simulated annealing. If you don't know what that is, that's okay. What we say is that you should specify this if you have a single chromosome or a simple model, and if you have a complete genome or a complex chromosome model, you can skip this altogether. If you skip this, then we have a Metropolis Hastings simulation, and that works better for more complex systems. Then this is the input GTrack file to Chrom3D, and finally the output will be sent into this file called model.cmm. You will see from Chrom3D that there is some output information as soon as you start the program. It will say, that, for example, that it rescaled the bead sizes from 0.0, 0.7 and so on to 0.01. That's just the volume that we set here. We said 1% because in the GTrack file we specified a radius of 1 for each of the beads. This will be the case. It says we are looking at one chromosome only. There are 97 beads, 71 interactions between the beads, and there is some information here that we're not using. 31 of the beads should be restrained towards the periphery, and that's it. And then it gives the loss score during the simulation. So my simulation is now finished, and I have a model.cmm provided for me. You don't have to do this. I'll just show you what this is. A simple file providing X, Y, Z coordinates, radius, and RGB, so color values and so on, and bead ID. This file will, so the next step, we are actually soon now able to visualize this in the Chimera system. So what I'll ask you to do now, so I'm sitting on a server here, so I will need to copy this file to my local computer. So what I will do, I'll use SCP and basically download the file to my local computer. If you're on PuTTY, you can, I guess you can drag and drop the file to your local computer. I haven't used PuTTY, so I'm not sure. If you run this on your own computer, that's of course also perfectly fine. You can just open it directly. So I think I'll open the model.cmm in Chimera X now. If you're on a Mac like so I've transferred this model.cmm from the server that I'm using to my local computer, which is a Mac, and I've properly installed Chimera X. So what I can do is just type open model.cmm. Yeah, sorry for that. There is a dot missing here actually. So if you have problems here, you should do copy model.cmm with a dot here so that you actually copy it correctly. So there's a dot missing here and here. So please, if you need to copy, you should add this dot here basically. Sorry for that. If you're on a Mac, you can type open model.cmm. On other systems, I guess you'll have to open Chimera X and then use that system to select the model.cmm file for you. But I'll just use the open in this case. Then you can see hopefully something like this. This is now a visualization of the chromosomal model. In itself, it doesn't provide so much information, perhaps. It gives you the overall structure of chromosome 18, but perhaps not much else. So what I usually do after running such a model is what I've also provided the information in step 18. There is a command here that you can copy. This is a Chimera X command listed here under the third bullet point. There is a command line interface down here for Chimera X where you can paste this command in. And what this will do when I hit enter is that it'll make a sphere which represents the nucleus. So this basically sets a nucleus centered at the origin with a radius 5 and with a certain color. Of course, in itself, this is not very handy. It's just a ball. And now our chromosomal model is stuck inside the ball and we cannot see it. So the next thing to do now is to go down here. You can see Chrom3D model and you can see sphere here. So where it says sphere, you can click on the color and of course you can change the color, but you can also change the opacity of the model. For example, you can set it to around 30%. And exit. So now we can actually see where the chromosome is placed relative to the confinement of the nucleus. Another thing that can be handy is which I also provide information about here, is you can use a command called clip, which will clip this nucleus and just make sure that you have the chromosomal model facing away from you. So it's kind of not like this, but facing away because it will clip in your visual plane. And you just type clip, and then you can actually also kind of look inside of the nuclear model to see the chromosome placed there. You can also play around with, if you click full, You will see some fancy shadows and things like that. You can use the soft. I also think the soft looks pretty good for visualization purposes. And then, yeah, so this is usually a good way of visualizing if you have a single chromosome that you want to visualize. Then, of course, what you see from this is the information lacking from here is actually, we can see some of the beads being placed, sort of embedded within the nuclear, inner nuclear membrane here. But how do we know that these are actually the LADs? So how can we, all of the beads are colored in the same color now. How do we know whether these beads are the LADs? So basically, how do we know that the LADs are the beads placed in the periphery? So that's to determine or to visualize that, we can go to the step 19, which again, going back to where you ran this and copy the commands under step number 19. This unzips another script that we're going to need, which is called color beads. The color beads script is another Python script that will allow you to color the beads by providing a list of bead IDs. So basically what we do in this step is selecting from the GTrack file all of the beads with a number 1 in column number 6. So this is the periphery column of the GTrack file. Each of the lamina associated beads, we fish out the ID and put them in this file here called LADs IDs. These are IDs of all the LADs. Then we use this script, color beads, which takes in the model that we generated. It takes in the IDs of the LADs and specifies using RGB a color. So this will be red because you have 255,0,0. And the keyword override just makes sure that any color you already have in your model will now be overridden by the colorization that we are specifying here. And then it will output that into a new file called model redlad.cmm. Again, this one you can download to your local computer. Important question. What can be the reason for the model being slightly different from the model I'm showing? So there is a random number generator being used as part of setting up the chromosome and of course randomly selecting the moves. Since we are sitting on different computers, different systems, the random numbers that will be generated will be slightly different. So for example, you will experience perhaps that I have a blue color on the chromosome. I didn't specify any color, so Chrom3D just selected a random color from my beads. So perhaps the color is different on your systems, or there are some slight differences in the structure of the chromosomes on your systems. So this is expected. This is expected and is also the purpose of going back to the previous question from some time back about running multiple models. So this is also the reason why we can run multiple models with different seeds each time to generate some statistics on kind of the stability of this structure that we're looking at. And the question about the random seed, it can be fixed. I'll show you actually how to do that. So what you can see, you can just type Chrom3D help and you will get a list of all the commands. There is a parameter called -s or -seed where you can set a number. It's by default just set to 1, 2, 3, 4. But if you select a different number, then the randomization will be different basically. So if you set, so for reproducibility, you can get the exact same structure by specifying a certain seed. Yes. So if you have now been able to download the new model, the one called RedLad, I will show you this one. Okay, I'll try not to look at both of them at the same time actually. So I'll just look at the model called red lad. This looks like this. You can see now some of the beads being colored red. I'll do the same as I did previously with making the sphere and selecting the opacity around 30%. I will also use the clip command so that we can kind of see better. And I'll use the soft visualization. So now you can actually see the red beads are the beads where we had lads. So now you can actually see that these are the ones placed towards the periphery of the nucleus as we expect. And the gray beads are all the other beads, and you can see them kind of facing more the nuclear center. Overall, the chromosome is positioned in the nuclear periphery. That's known for chromosome 18 in this system. But I remind you that we are looking at a rather artificial system here, since we are only looking at one chromosome. And of course, there will be, from the Hi-C data, lots of interactions between chromosomes that will also be able to kind of specify the positioning of these chromosomes. Finally, I can also show you what we often do if you want to put this in a publication. Sometimes it can be good to have a white background, but what you will see is then it looks a bit strange. So what I usually do then is to reduce the opacity, or increase the opacity a little bit. And also there is a command, it's listed also in the tutorial, where it sets the depth Q color. So now the depth is indicated with a white color, but if you use black, it will be more like a kind of shadow, like you can see here. Of course, you can also specify a different color or a different opacity according to what you think kind of looks better. For some final remarks, as we have touched upon already a couple of times, what we do in practice is to generate multiple models, starting with different random seeds to actually be able to generate some statistics from these models. So for example, in the publication from where Chrom3D was published back in 2017, we could, for example, generate statistics like this, where we'd show the distribution of the percentage of the TADs placed at the periphery if these TADs are associated with one of the components of the nuclear lamina, the laminae C, or all of the beads that are interacting and where they, on average, are placed in relative to the nuclear periphery. We can also generate visualizations like this, where on the y-axis here we have the different chromosomes, and here we have the chromosomal positions, and then we can color each of the beads according to how frequently it's placed at the nuclear periphery, for example. So with red color indicating a high frequency of peripheral location across, let's say, 1,000 models that we have generated, and then blue indicating that these regions are more stably placed in the nuclear center. But you will also see that there are several regions in yellow here which will sometime be, or in some models will be in the nuclear periphery, and in other models will be placed in the nuclear center. Yes, as I already mentioned, there is a full genome version of this INC tutorial that you can access. Here, if you just go to github.com/chrom3d, you will see today's tutorial, but also a tutorial called INC tutorial full genome that you can try out to generate a full genome model. This we have also touched upon. What if I only have Hi-C data? I don't have any information about LADs. You can simply skip this particular column in the GTrack file, and it will, then Chrom3D will understand that you don't have LADs, and that's also possible. If you want to read more about running Chrom3D, we have a Nature Protocols paper that was published in 2018, where we also go through and explain the steps needed to run Chrom3D. But in the tutorial today, I showed you how to go from the raw data all the way up to the model visualizations. Yeah, if you try this yourself and encounter any issues or problems, please post these as issues on the GitHub page. If the problem is related to the tutorial that we have today, post them on this GitHub page. And if you encounter problems with Chrom3D, you can use this GitHub page and post an issue there. So we have a question in the chat, whether it's possible also to define the location of the loops in the input file and not just the TADs. So the way it works is that you can use any kind of segmentation on the chromosome. It's up to the user to define in which way this is useful. And this also includes the interactions between the regions. So if you have a way of defining loops, let's say by making a small bead, for example, and an interaction with a small bead in between several ads, that's certainly possible. But the way it works is that it's up to the user to define how the chromosome should be segmented and how the different regions should be restrained relative to one another and relative to the nucleus, basically. Another question is if it's possible to run this without any spatial indicator, like the Lamin V1 ChIP-seq, and that is possible. And I get that question quite a lot. So what if I don't have LAD data at all? Then you basically skip adding that column in the GTrack file, and Chrom3D will then understand that I should not care about the nuclear confinements, basically, and it will only focus on the Hi-C interactions. So that's certainly also possible.
Is Chrom3D strictly used for eukaryote, or it can be used for prokaryote as well?
If you don't specify any information about a nucleus, there will be no nucleus in the model. So in theory, it should be possible. I haven't tried it, but I see no, at least off the top of my head, reasons why it shouldn't work. So I think it should work. If you don't specify any constraints that has to do with the nucleus, there will be no nucleus that you will use for the modeling. Yes, there is another question regarding TAD calling. As I think I mentioned, you can use basically any TAD caller. You can actually do the step that I showed you here, but with another TAD caller, just making sure that you use this bed tools complement to fill in the gaps in between the TADs. Then there's a question regarding the best practice for calling Chrom3D without the use of LAD to include GTrack column and set all of the rows to dot. This will be identical to excluding the column altogether. So there is no difference in the behavior of Chrom3D whether you give a dot and still include the information about the periphery, or if you just exclude the column completely. So there is no preferred way of doing it. Either or will work in the same way. We have another question, which is about how does the information of the statistical significance of the interaction count map into the model's restraint? So the way it works in how we're going to do it today is that we have an either or type of situation where you can, so if you have a significant interaction between a pair of beads, then they will be pulled together if it's possible. And if there is no significant interaction, they will not be restrained to one another. I showed you that it's possible to specify extra information about the interactions. So we are not going to do that today, but it's possible to modify it so that if you want to prioritize interactions that have a lower p-value, or you want to prioritize interactions with a higher log-fold difference in the observed versus the expected. That's also possible to do in the Chrom3D specification that I showed previously by adding weights to the interactions. But that is not something we are going to cover today, but it's possible to do so. I see there is a question already about whether it's possible to superimpose different models to compare. So if you mean in Chimera X, this is possible by basically opening. So whenever you open a new model, it will appear down in this list you have at the bottom here, and you can turn off and turn on the visualizations of the different components here. So if all of them are on, you will be able to superimpose them, if that was what you meant. Yes. Okay, so now we have a question about how we define this restraint. So during the restraint creation process, are you using a power law to convert interaction frequencies to distances? No. In today's tutorial, the Hi-C data was used to determine statistically significant interactions. And for all of the statistically significant interactions, we set a constraint to minimize the distance between these beads. So there is no distant restraint at all in the, we don't assume that higher number of interactions means actual close, that they are closer in 3D space compared to an average number of interactions. We just say if it's a statistically significant interaction, we want these beads to be close together. And then there's a question about using other TAD callers. Yes, this is possible. I think I've touched upon that previously, but you can use any type of TAD caller. Just make sure to use this tools complement program to make sure that you have a fully segmented chromosome model. Basically, all the chromosomes need to be filled by segments. One, can we also integrate compartmentalization A and B? So one thing that we have done in some systems where we don't have, for example, LAD information available, is that you can use compartments as a proxy for LADs. So it's known that the relationship between compartment B and LADs is rather high. And it's also known in general that compartment B is more positioned towards the nuclear periphery, and compartment A is more positioned towards the nuclear center. So if you don't have LAD information available, you can use compartments as a kind of proxy to indicate positioning towards the periphery and towards the center. And that works remarkably well also.